in photosynthesis, you need to understand these two graphs, the absorption and the action spectra. So let's have a look at what these are. So first of all, we need to discuss an experiment done by a man called Engelmann. And basically what he was trying to find out was whether different colours of light affected photosynthesis differently, because he used his understanding of the fact that white light was made up of lots of different colours and that green things would reflect green light. And so he was sort of proposing that different colours of light may well have an impact. So what he did was he put green algae on a microscope slide, and he then covered the rest of the slide with bacteria that required oxygen to respire. So the bacteria were going to move to where there was lots of oxygen. Where there was lots of oxygen is where the algae is photosynthesizing the most. And then what he did was he shone light through a prism so that then there was the rainbow on the green algae, as shown here. So what you can see is that we've got the white light being shone through the prism. And so then you've got all of the different wavelengths of light here hitting the algae with different wavelengths. And what you see is that the bacteria are making a peak where there is blue and purple light, and then they're making another peak where there is orange and red light, and there are very few bacteria here in the middle where there are green light. And that allowed us to develop what was called, start to develop these graphs. So the first graph is called the absorption spectra, and basically this shows how much light is absorbed at each wavelength for each of the main pigments. So we've got chlorophyll A, chlorophyll B, and then the carotenoids, which are the accessory pigments. So they're the ones that you really start to visualize when the leaves change color. And what you can see is we've got a peak in the blues and the purples, and then another peak in the oranges and the reds. Attached to this is something called the action spectra, and what that is again looking at with the wavelength on the x-axis. It then looks at the rate of photosynthesis on the y-axis, and you can see that you've got a peak here which correlates to the purples, and you've got a peak here which correlates to the reds. Now perhaps the most interesting thing occurs when you put both graphs together, and hopefully what you notice is that if I were to draw a kind of an overview, like almost like an average line for the absorption spectrum, it pretty much exactly matches the action spectrum. And so from this we were able to conclude that it was the chlorophyll pigments that were involved in photosynthesis. Now you do need to be able to draw these. You need only for chlorophyll, you don't need to add the carotenoids, and you need to be able to label the x-axis, even if it's just with the colours rather than with the numbers, but you need to know that it's basically going from purple through to red.